Fantastic. Thank you very much for the introduction. Yeah, so this is a partnership that's about a year and a half old that is seeking to create new treatments, possibly even cures, for genetic heart muscle diseases. These are conditions that a cardiologist would call cardiomyopathy. There are different types, but you don't really need to worry about these distinctions. They all have in common that if your heart is affected by one of these, you are at risk of progressive heart failure, or you are at risk of intractable, dangerous rhythms that can cause you to drop dead suddenly. And if those things happen, we don't have any disease-modifying cures. We could do a transplant, we can put in a defibrillator, but we can't fix the underlying problem. The unmet need is huge because these conditions are surprisingly common. So we have confidence in this number, 1 person in 250. Completely different scale from most rare genetic disease. And that gives opportunities, but also challenges. It's perfectly likely there's somebody in the audience today who's got one of these conditions. Fortunately, they're not always serious, but they certainly can be. And because they affect relatively young people, they're the main cause of heart disease in young people. They're responsible for about half of all heart transplants. And if you hear about a sports person collapsing and dying suddenly on the pitch, this is probably what they will have. So they drive very major healthcare costs, and that will be important in terms of trying to position therapies. There is a very substantial monogenic component. It varies in terms of the proportion of the disease and the different cardiomyopathies, and the different genes vary, and I'll touch on some of them. Maybe broadly 50% is monogenic, in which case it's autosomal dominant, adult onset. There are also polygenic forms, but the severe end of the disease, sorry, I've gone backwards, the severe end of the disease is this rare pathogenic variant, single variant, which you could address with a genetic therapy. So these are nasty diseases for families, and I'll illustrate that. This is the family of Wendy Borsari, who's a patient advocate in the CureHeart team. Dominant transmission, lots of black symbols, affected individuals, lots and lots of heart transplants in this family that have hypertrophic cardiomyopathy. Heart transplant's not a great thing, doesn't always go well. Lots of young people dying suddenly. So a nasty disease, but not as relentless or aggressive as many of the conditions we've heard about in other sessions. These are diseases that play out over many years or sometimes decades, and that's one of our particular challenges. So we know a lot about the underlying biology. This is a slide relating to hypertrophic cardiomyopathy, which is the most common one. The mutations here affect the motor proteins of heart muscle. This is a sliding filament model. And it turns out that the reason these are common is that the genes here are big structural proteins that make very big targets for mutations. They're often missense. Many different missense mutations can mess up this motor and cause disease. For hypertrophic is actually a gain of motor function. So we know a lot about the biology. There are some small molecule drug developments. There is a class of new drugs, the cardiac myosin inhibitor, that was built on this work. There are other molecules that have been repurposed to try to improve the problem, which is one of inefficient energy usage. They improve symptoms, but they're not disease modifying. So the question has always been, if you know the precise mutations, why are you not deploying genetic therapies? And when we started thinking about putting the CureHeart program together 2 or 3 years ago, this was happening, as you heard, in liver, in bone marrow, in retina. Heart was in the too hard pile. But actually, I think there are some advantages in heart. The heart is a post-mitotic organ. If you can edit the cells, your edit will be lifelong. It has very little or no cancer susceptibility. And cardiologists are quite good at getting things in the heart through the vasculature. We know how to do that. So there are challenges, but perhaps opportunities. But it wasn't happening. And the thing that changed that for me, and I think that's important for this partnership topic, was a very bold program initiated by the UK heart charity, the British Heart Foundation, who decided for the first time they were willing to fund globally and willing to put out one large award for anybody who could convince them they could make a transformational change. And I think that was the key that opened the lock, that it enabled us to put together a team drawing on whoever in the world you would most want to work on this challenge. And so the team we put together, and I lead it, called CureHeart, was fortunate enough to win this Grand Challenge competition from the BHF. And I think the reasons we were successful is partly we could get a really great team together, and I will briefly introduce them, but also because the timing was right. We had the building blocks that we needed. So we have 3 of the laboratories, including myself, are those who worked on the underlying genetics, the gene mechanisms. We can find the patients, we can find the families, we know whether we're talking the dominant negative or haploinsufficient gene. But this is the only bit of the program that's cardiology based. We then have colleagues, some of whom you will know, who have created the editing tools that we aspire to use. David Liu, I will talk about with base and prime editors. Eric Olson, some of you will know, has been pioneering CRISPR editing in Duchenne. Targeting the heart is, of course, hard. Luke Vandenberg, very involved in AAV9 development. Matthew Wood, my colleague here, who's put together this meeting, brings RNA biology and delivery expertise to muscle, and John Simon made most of the models that we all use. So it's a really exciting team to work with. It was meant to be a grand challenge, and we do have some very significant challenges. One is the clinical piece that I spoke of, that the time courses are quite long in this disorder. The other is the nature of the mutations is somewhat challenging. So our patients are always heterozygotes. They have a wild-type copy and a mutant copy. For most of the genes and most of the variants, these are dominant negatives. It turns out the genes in which the dominant negative mechanism causes disease are not dosage sensitive. And so actually we have the opportunity of either correcting or silencing that copy. But there are other genes, including 2 of our very dominant, important ones, where the problem is loss of function and haploinsufficiency, so that's a gene supplementation or upregulation approach. For both of these, we are developing both RNA therapeutic reversible strategies, but also moving towards editing strategies, which should be where we can begin to talk about cure. For the gene supplementation, clearly, if the gene that you want to put back is a small one, you have the option of doing a conventional genetic therapy. It's not what CureHeart's doing, but I will show 2 slides on this because it is happening and it's very relevant to the field. So a number of companies, and this is just Tenaya, who's one of a number, have looked at a number of cardiomyopathy genes where the gene product is small enough to go in an AAV. And these are either just doing the dose ranging study or getting ready to do the first studies in man. So at one level, this is very exciting for the field. But I think there are some significant caveats here. And I will illustrate that by this Tenaya study. So you won't, if you're not a cardiologist, know what this means, but this is the entry criteria for a patient for this AAV9 gene therapy. And this describes a pretty typical patient. I will see patients like this every week in my clinic. I could find at least dozens of patients who meet this criteria within my practice just in Oxford. And they're broadly fairly well. I would expect them to be well over the next 10 or perhaps 20 years. So they're not like a boy with Duchenne's or a baby with no... I'm blanking on the protein, but I've just heard the lovely lecture on surfactant in the lung. These are relatively well people, so our tolerance of risk needs to be a little low in this circumstance. And as you will know well, AAV vectors have acute immunotoxicity problems, which may be a bit more marked in someone with a diseased heart, because myocarditis is one of the problems, and our patients already have myocarditis. And even before we get into the idea of using a viral vector for an editor, which will have additional immunotoxicity problems. Overexpression is not necessarily innocent. We could talk about that. Will you get durable expression of the transgene for the life of the patient is unknown. And this was touched on earlier. This is a dose-finding study. If you take part, you will make neutralizing antibody. You can't then take part in a version 2 or version 3. And I think that's a real problem for informed consent. So in the upregulation space, this is not what we're doing. we're doing work, which I won't talk about today, looking at manipulating transcription and translation to try to increase production from the wild-type allele. But I will tell you a little bit about some of the dominant negative efforts. The problem that we have here is one that you will have spotted. We have multiple, many different missense mutations. In my schematic, these are the 2 parental copies of a myosin gene. This is a missense mutation that causes hypertrophic cardiomyopathy. This is a different missense mutation that causes dilated cardiomyopathy. They actually have opposite biophysical effects. It would be relatively easy to design a strategy, for example, a base editing strategy, to correct this. But if there are only 10 or 20 or 30 patients with that variant in the world, it's not N of 1, compassionate use, these are fairly well people, and it's not a big enough number you could do a specific trial. We're not confident that that is a feasible strategy. So we are therefore pursuing a strategy that has also come up today of saying we don't need to target the rare variant, we just need to discriminate the maternal and paternal copy. There are common tagging synonymous polymorphisms, usually the 3rd base pair of codon, that are not selected by evolution. They're really common. You can find some of these that have a 50-50 allele frequency. So if you create a silencing tool that can discriminate the C or the T, you show them to be effective and safe, you put them on the shelf, 50% of all patients who turn up with a mutation in this gene, you can treat with one or other of these agents. So this is a strategy that we think can be commercially viable. If you have to be very selective to discriminate on the basis of a single nucleotide change, can you be effective enough? Can you lower down enough to deliver what you need? Turns out you can. This is reassuring data that we've had for quite a long time. It comes from a mouse model. So the mouse has the mouse equivalent of one of the human missense mutations. The heart is thickened and it's scarred. So RNAi, AAV-delivered RNAi, that targets the missense mutation, not actually on the basis of the missense allele, but actually a tagging SNP, delivered early in the life of the mouse, provides a durable cure. That mouse remains free from cardiomyopathy through its life. And the headline finding that's important for us is that was achieved with relatively modest reduction of the mutant transcript. So we need to be selective, but we don't need to completely remove the mutant transcript. And there are reasons why that is the case biochemically, and we could talk about that. So we're interested in RNAi, we're interested also in ASO approaches. This is work that we do collaboratively with Matthew's lab, and because this is an enzymatic cleavage, there are opportunities here for finding ASOs that discriminate on the base of a single letter difference, which we heard about earlier, and we can work in human-derived stem cells and tissues and not have to do this in cell lines or mouse models, and we can make humanized mice. So these are approaches that I think are feasible and might be quite appropriate for many of our patients, particularly patients who have the less severe disease spectrum. Of course, we are interested in longer-term and more definitive treatments. We're very interested in editing. We're not specifically expecting to use CRISPR-Cas9. We don't want double-stranded breaks. The heart is post-mitotic. We can't do homology-directed repair. So this is why we have David Liu on board, because we're much more interested in base editors. And these have had coverage today. Astonishing piece of chemistry to create an enzyme. There is no adenosine deaminase in biology, but David can use directed evolution in the lab to create these base editors. And of course, if you know that C to T is the commonest human mutation, being able to reverse that is extremely useful. Base editors, as a proof of concept, work beautifully in this setting, and we can use them to fix that same mouse model. So this is a similar experiment. This is the untreated mouse. This time it's dual AAV. It's a big cargo, so there's a split-in team to bring them together. It's an AB8. And it targets and corrects the underlying mutation, and if delivered early in the life of the mouse, produces a cure. So it's lovely proof of concept, but you will spot that there are some issues here. One of them is that this is actually correcting the mutation rather than silencing, which is a strategy that we think we will need to develop to treat enough patients. And the other is, this was achieved by very, very early delivery in the life course of the animal. We do have a nice window on our patients. They tend to be healthy in childhood, usually in adolescence. So we've got maybe a 10-year window, maybe 15. But if you start doing treatment once the disease is advanced, at least in the mice, we're not sure we're going to get a whole lot of reversal. You might stabilize disease. So the timing window is important. So just to put this in context and to wrap up, whether we're talking about the reversible approaches, which I think is what we anticipate will happen first in our work and perhaps others in the field, and then getting on to editing, obviously this is only somatic. You'll understand why. We've got very particular issues about risk and balance. We've got very large numbers of patients who have moderately high risk of morbidity and mortality over 10 or 20 years. But that's a somewhat challenging thing for case selection and designing studies. We have a clear tension that if we are to deploy these therapies early in life, we're much more confident that we can achieve a marked effect and perhaps a cure. But I don't imagine that's where trials will start. Much more likely we'll have to start in advanced disease, quite possibly people who are on a transplant waiting list. And then we get the heart out, we can see what we've done. But it will be much harder to achieve benefit in a heart that is scarred. And then obviously, if you can do this for rare disease, any gene expressed in the heart becomes a target. So I don't have a credit slide that details all the individuals, including in my lab and Matthew's, because this is a very big program, and instead just the headline of the scale of the team that we brought together to deliver this, and a big shout out to the British Heart Foundation that put up the money and made this possible. Thank you.